So today we are going to do one important practical on an sophisticated analytical instrument, that is HPLC. Full form of this instrument is high performance liquid chromatography. Okay. So what is the major uses of this instrument is the qualitative and quantitative analysis of the samples. So separation of the component from the mixture of compound. Okay. So this instrument having few major equipments or component, like that you see, first one is the solvent reservoir and the degassing system. What is solvent reservoir? Solvent reservoir is the container where the mobile phase is kept, which can be used during the separation. Okay. So this, from that solvent it is coming inside, and before entering the instrument it is passing through the degassing system, that is the purging system. There are different degassing system like distillation, ultrasonication, and the purging system is also there. So this is done instrumental. So in purging, what we do? We remove the dissolved gases from the mobile phase. If any gases are there, it will be removed from this portion. Okay. So after that, the cleaned or the refined or portion of the mobile phase is entering inside the instrument. Now you see, next another important part of this instrument is the pump. Pump is the very important component by which the mobile phase is thrown or flown in a very high pressure and very high flow rate. This is the pump of this system. Here is the pump is kept generally. Okay. So once sound is coming, that is actually this we are using in this sophisticated instrument. We are using reciprocating pump. The sound is coming from the reciprocating pump, because in reciprocating pump pistons are moving backward and forward. That's why the sound is coming. So pump helps the mobile phase to enter into the column in a very high flow rate. So next portion is the injector. What is this injector? You can see here this injector, and it is having the two knob is there and two position is there. One is the load position, another is the inject position. So what is this? How the sample is injected? So we are using this syringe. This is a specialized syringe used for the sampling or the injecting the sample in the instrument. I can see the needle is blunt, not the sharp like human uses. So what we use, we take the sample inside this syringe, then where we keep the sample or we place the sample inside this probe. There insert probe is there. Through the insert probe, we insert the sample in here, and generally the knob is kept in load position. What is load position? Load position means sample is loaded. Whenever we are transferring the sample from the syringe to in this position, the sample is loaded. Next, when we move the position from load to inject, then actually the sample is injected in the system. That means the sample is going inside the column. So this is a very crucial part of this instrument, is the injector part. So here the sample is introduced or injected in the system using this knob, which is the load position and inject position. And this type of injector we are using, this is known as rheodyne injector or loop injector. These are the loops. You can see the loop. This is the loop, they are containing generally 10 or 20 microliter volume. This loop, the sample whenever we are loading, the sample is stayed in this loop. And when we keep this position into inject position, from this loop they go inside the column. Next part is the column. You can see this is the HPLC column. So this is one of the very important part. This is the C18 column. What is C18 column? C18 means it is also known as ODS column, octadecile silent. This is a silent type of column. That means here 18CH3 group is attached with silica, bonded with silica. Okay, so this is a non-polar type of column. Generally they elute polar component first. So that's why it is known as C18, because 18 CH3 groups are attached with silica. So this is the very important part, and this is the outer coating material made up of steel, and after this column Actually the separation takes place inside the canal. This is very important part of the instrument. Okay, this is also known as the heart of the instrument. So after the separation takes place, the separated components are going to the detector. So this is the last portion. There is the detector part. Generally we are using either UV detector or PD detector. So here in our instrument we are using the UV detector. So in having the variable wavelength, we can set any wavelength. So we can analyze the sample, we can scan the sample in any lambda max. Okay, so this is the detector part. Sample is detected and ultimately the component is, this is the recorder or the software. So we are ultimately recording everything inside this recorder or the software system. So that whatever the peaks we are getting, we can store it for future, we can modify, we can edit it after using this software. And this totally instrument is operated by using the Agilent software. Our model is that instrument we are using Agilent model 1920 model is there, and this is totally operated by software. So we are using the injector, whatever the basic application now if we are discussing. So we can use qualitative analysis and quantitative analysis. What is quantitative analysis? If we perform any type of assay of any drug or any molecule, that is known as quantitative analysis. We can determine the percentage purity, and also apart from that we can go for separation work also, that is qualitative work. If we are using a mixture of sample, maybe a natural product or any other synthetic product from a mixture of product, if we are separating the component by using this instrument, that is known as qualitative. Okay, so this is a very sophisticated instrument, and you can see ultimately the whenever the mobile phase is running, pump is running, so mobile phase is coming out eluted and it is stored in the external reservoir. This is one external reservoir for the storing that eluted mobile phase. Okay, so that's all for this demonstration. Next we will perform the experiment after that. Okay, thank you.
Sir, one question. Which software is used here?
Recording.